All right. So today let's talk about cell structures and cell membrane. It's one of the most fundamental concepts in biology, and we'll get started from here. Um, Today we'll be addressing three major types of cells: prokaryotes, which shown here is a bacteria, prokaryotes, and the two types of eukaryotic cells. Let's talk about how we would group these cells. Prokaryotes and eukaryotes are grouped according to whether they have a nucleus at a certain point of their life. You can see here that the bacteria cell does not have any nucleus. This is no nucleus, or you can say a nucleus. While both these two cells have nucleus. The nucleus of this one cell shown here is over here in the circle, and this one shown in blue. These are both nucleus. All right, and we'll talk about it later that this is a animal cell, and this one is from plant. All right. Before we begin talking about all the cell structures, let's address the cell theory. The cell theory is basically um three principles. First one saying that all living things are composed of cells or cell products. That means everything in your body that is responsible for the functions and for you to be alive, they're composed of cells. Your heart, your different organs, your skin, your eyes, your mouth, everything, they're composed of cells. And the cell is the smallest unit of life. That means any subunit within the cell cannot support a life by itself. And the cell and each one of your cell is capable of supporting the life. The third principle is that cells only arise from pre-existing cells. You might have heard of mitosis, which is how cell divide itself in humans. Um, you might have heard of binary fission. That's how some prokaryotes that's how they divide the cells. In prokaryotes divide themselves and create more. You can't just have a cell emerging from nothing. It has to come from a pre-existing cell. Okay. Um, and after that, we already did the cell theory principle. Next, we'll be talking about the cell structures of the three types of cells that I mentioned before. Um, Prokaryotes, animal cells, and plant cells. These three. And after we've done all of those, we'll compare and contrast the cell structures of prokaryotes and eukaryotes. This is really important as a lot of IB questions actually address on comparing and contrasting. Let's look at prokaryotes. Um, can you briefly point to me which components or structures do you find interesting or do you think it's important in this cell? Yeah, so this long tail is important. These small hairs are important. This one here, it looks like a clump of strings. It's important. These small dots are also important. And that little circular thing, which is actually DNA, is also very important. Also, the capsules, the three layers that covers up the whole cell, the three layers, they're also very important. So let's look at them. First of all, the long thing that looks like a tail, that's called a flagella. Flagella is a type of microtubule, a microfilament, microtubule. Um, they are in charge of movement. All right, they're in charge of movement. And the small pili, which are shown here as the short green hair, they are responsible for anchor. So it helps the bacteria. This is the bacteria cell, right? It helps it to stick to your cells. Imagining there's like the lining of your small intestine, and the bacteria will be able to stick to it tightly with the little hairs. So we've done flagella and we've done pili. Next, let's look at the capsules. Okay, the three layers that covers up the inside of the bacteria. The most outer membrane or layer, it's called a slime capsule or capsule. It serves the purpose of protection. And the second layer is in the middle. It's called cell wall. It's similar to the cell wall that plants have, but they're composed of different materials. And the third one is the cell membrane, the green one, the thin one. This one is also common in animal cells, the cell membrane. Um, but cell membrane is the only membrane that human cells or animal cells have, while bacteria actually have three, which makes them tougher, I guess. The next slide sums up some of the key features of the bacteria. Oh, we forgot to talk about the inside. We forgot to talk about it. Um, the intracellular fluid is called the cytoplasm. It's called cytoplasm. It's basically the aqueous environment for all the organelles and the cells to function. The little dots are called ribosomes. This is a ribosome. Um, it kind of looks like a hamburger if I draw it simply. It has a smaller component and a large component, and they're in charge of the production of proteins. Okay, but that's not important at the moment. We'll talk about it in future lessons. Also, we have the plasmid and the nucleoid. Okay, plasmid and nucleoid sums up the whole genome of the bacteria. Okay, they're DNAs. Both of these are DNAs. But these are really different from eukaryotes, which we will show later. The eukaryotes store the DNA in the nucleus, which has a membrane covering for it, while the bacteria has a naked nucleus. And the space, the imaginary space, which has the DNA in it, is called a nucleoid. It's called a nucleoid. And this one, this plasmid, it's a little circular DNA. It codes for a lot of different things, and they're really important for the bacteria to actually infect and prevent the immune system to destroy other bacteria. The plasmid is actually also responsible for fighting off viruses. It's a competition, right? And this one just emphasize on some key features. The cytoplasm is the internal fluid component. We've talked about that. Nucleoid, the region of cytoplasm where the DNA is located. We also talk about that. The plasmid, the circular DNA, and the ribosomes, they're complexes of RNA. Actually, RNA, my bad. And proteins, they're responsible for polypeptide synthesis. That's that. And about the 70s and 80s, we'll talk about that later, but there are two types of ribosomes. One is 70s and one is 80s. Some slight differences. Okay, now let's get to the animal cells. The animal cells are more complex, as you can see. You got more organelles, and they're called the membrane bound organelles. which means these organelles have their own membranes. Now, let's look at this from the inside to the outside. The most important part, I guess, in a cell is the nucleus. It contains all the genetic information. And this that looks like a kind of an egg is the nucleus. And among the nucleus, you can see the little holes on it. I use a different color. The little holes. These are nuclear pores. They allow materials from the inside of the nucleus to travel out into the cytoplasm for further process or functions. Within the nucleus, you can see the nucleolus. Nucleolus, what is nucleolus? It contains RNA. It contains RNA that is crucial for the cell function. If I draw out the membrane of the nucleus from a top view, it kind of looks like this. These are the pores and these are the envelope. Why do I draw it like that? Because a nucleus is a double membrane, has double membrane. Okay. You got two layers of membrane. Um, one is here and one is here. Because it got nuclear pores, so I cut them off at here. And within the nucleus, there are your DNAs. This kind of looks like the nucleoid of the bacteria, but they're covered within the envelope. Also, there's this nucleolus, which contains RNAs. And around the nucleus, you can see these green things. They're called the um, endoplasmic reticulum. Endoplasmic reticulum. And there are two types of endoplasmic reticulums. Yeah, one is smooth and one is rough. From the from this diagram, you can probably tell that the difference between smooth and rough, they got these little things on the rough on the RER. These little things are ribosomes. And do you remember what ribosomes do, as I mentioned when we talk about bacteria? Yeah, they are responsible for protein synthesis. So RER is responsible for the production of proteins or polypeptides, and the smooth ER is responsible for the production of actually lipid and something else that you probably don't know at the moment, and we'll talk about that later. But now, just as a little intro, SER is for lipid and RER is for protein synthesis, okay, polypeptide. Okay, now let's move on to other organelles. You can see the orange thing here. You probably learned in probably like middle school or primary school, mitochondria are the powerhouse of your body. But now you need to know a lot more than that, okay? But still, they're powerhouse of the body, which is not wrong. They are responsible for producing ATP, which is the energy currency of your body. And same here, mitochondria. If you look at the membrane, you got two layers of it. One layer is the outside, this one. The other layer is the inside. That looks a little different. They are folded. They're folded. When the membrane is folded, like in your small intestine, the lining is also folded. Um, Realize? Can you imagine or can you think of one of the reasons why the inner membrane needs to be folded? Yeah, to increase surface area. That's right, to increase surface area. So the outside, um, of the mitochondria is flat, but the inside of it is folded to increase the surface area. The main function of mitochondria is to produce ATP, and about the details of mitochondria that we will address later in another lesson because I only got 200 minutes. And now this purple thing is called a Golgi apparatus. It looks kind of similar with the ERs, with the endoplasmic reticulums, but it's very different. One major difference is that you can see all these um, 3D tubes. They are actually separate from each other and not interconnected, while these ones you can see they're connected among each other. And Golgi apparatus, the main function of it is to process and package the proteins. And the detail of that we'll talk about it later. It's not something we can explain in a few sentences, but basically, when the protein needs to be produced, a certain section of the DNA within the nucleus will first be transcribed into a messenger RNA, and that messenger RNA will be passed onto the RER. It's a little hard to see. Passed onto the RER, and they be processed into polypeptides. And that polypeptide will then be passed on into the Golgi apparatus, be processed and packaged into mature proteins. And they sometimes the proteins stay in the cell, sometimes they get delivered out to the cells into other parts of your body or another cell. Okay. Now, the key components of the animal cell, you got the nucleus, you got the mitochondria, which are the powerhouse of the body. You have the Golgi apparatus, um, and the endoplasmic reticulum. Ribosomes, there are ribosomes fixed on the RER, but there are also free ribosomes. Sometimes you might have to differentiate those two. Um, but yeah, these ribosomes are ADS, different from the prokaryotes. So that's something important, and we'll talk about that later. The cytosol is the internal fluid of the cell. The all the green stuff you can see, not green, all the blue stuff you can see here are the cytosol. They're just into liquids of the cell. Also, I forgot to mention the cytoskeleton. Um, the cytoskeletons are the strand you see here. It's just a really simple diagram, a representation of the cytoskeleton. It's actually way more complex than this, but you just need to kind of know the cytoskeleton holds your cell in place, in shape. Otherwise, it'll be all squabbly, but they act as a backbone of the cell and holds it in shape. It's a support, okay? Now let's get to the plants real quick. Plants, most of the plants organelles are talked about, we mentioned in the animal cells, but there are certain unique um, features of a plant cell. Well, quickly, there are the nucleus and there are the nucleolus, um, which is the same in animal cells. There's also the rough and smooth endoreticulum, so sorry, RER and SER. We also talk about that in animals. The free ribosomes, ADS, we talked about that. Mitochondria, which creates ADP, we also talked about that. Okay, and the Golgi apparatus, we also talked about that. These are all the same from animal cells to plant cells. Okay, maybe the cytoskeletons are also the same. Okay, what are the differences? You can probably tell there's something that you haven't seen in animal cell, right? One is chloroplast, and the other is the central vacuole. And we'll talk about the cell wall a little bit later too. So chloroplast. Do you know how humans acquire the nutrition and energy it needs? It's by respiration, okay? We eat food, we digest them, and the nutrients and all the organic molecules can be used by our cells to produce the energy we need. But for plants, there's another way of them to acquire nutrients or energies, is by sunlight. And chloroplast would convert sunlight or light energy into chemical energy that the plants can use. The detail of chloroplast and photosynthesis is very complicated, but we'll talk about that later, okay? We'll make sure that you get through all those. Um, Another difference is the central vacuole. It's basically a large pond of liquid that can adjust the size of the plant cells. Also, plant has to store a lot of water, as you know, and the central vacuole also serves a purpose of that. Then we'll talk about the cellular cell wall. Even though the prokaryotes, the bacteria also have cell wall, you can see that their cell wall is made of peptidoglycan, while in plants, the cell wall is made of cellulose. It's kind of like these pentagon things holding hands to each other, and they form a more rigid um, structure than the cell membrane. And the main function is to support and to protect the plant cells. Okay. Um, the key features of a plant cell is the central vacuole, the chloroplasts, which are responsible for producing energy, the cell wall, the cellular cell wall, thicker than a membrane, yeah, obviously, and the shape. The shape of a plant cell comparing to an animal cell, you can see it's got a more clear edge, I think. Um, while the animal cell is more like a round and it doesn't have an explicit edge or corner. It's because the plant cells have cell wall, which are more rigid and can hold them in place better. Um, I think we're running a little bit over time. Okay, so we'll finish by talking about the compare and contrast of the cell of plant of prokaryotes and eukaryotes. This I found from BioNinja, which is a really useful website where you can look for all the diagrams and explanations. I used it when I was in year 12 and it helped me so much. You can remember the main differences of prokaryotes and eukaryotes as DORA, DNA, organelles, reproduction, and average size. Okay, the DNA, the DNA in prokaryotes, the nucleoids, they're naked, no, there's no membrane cover for it, but this one is bounded. Um, actually, sorry, let me correct myself. Um, what this is talking about is DNA in prokaryotes um, are naked, like it's just a strand of DNA. But in eukaryotes, our chromosomes, um, or chromatins or DNAs, they're bounded to this protein called histone. So they're kind of packed um, onto a strand that helps fix it in place. Okay. Sorry about that. And in prokaryotes, DNA is circular, but in eukaryotes, it's linear. You've probably seen these ones. This is the condensed form of a DNA, but when it's loose, it's just like a strand of strand of thread. The prokaryotes DNA usually don't have introns. Introns are the part that you cut out. Actually, that is very confusing. But we'll talk about that later. And the eukaryotes also usually have introns. The organelles, obviously, prokaryotes have no nucleus, and that one has a nucleus. That's how we differentiate them. Um, the organelles in prokaryotes in bacteria is also not membrane bounded, while the eukaryotes are membrane bounded. The 70s and 80s ribosomes, that's something we'll also talk about. At the moment, I just need to know that about what the difference actually is. We'll talk about it later. And the reproduction of prokaryotes is by binary fission. It's kind of like it split itself like that, and then just becomes two. It's binary fission. And the eukaryotes are mitosis and meiosis. That we'll also talk about later. Okay. Um, Prokaryotes are mostly have single chromosomes. That means they're haploid. But eukaryotes, as humans, we're usually diploid or more. Okay. Um, you've probably seen these kind of diagrams with a lot of X on the screen. These are diploids. That means there's a pair um, of chromosomes, of homologous chromosomes in human bodies. And the average size of prokaryotes and prokaryotes, for example, bacteria, are usually a lot smaller than the eukaryotes. That's because their structures are more simple. Okay, I'll send the slide to you so you can have another look at everything. And if you have any questions, feel free to ask me. All right, sorry, I forgot to cover up the answers. Um, Do you want to have a go at this question and see if you can figure out the answer? Okay. Yeah, you're right. It's C. So I'll walk you through this question one by one. First one, A, cell wall are found only in prokaryotes. We know that's wrong because plant also have cell walls. But then look at B, naked DNA with histones is found only in prokaryotes. No. Okay, this one is completely wrong because histones and naked DNA they don't happen at the same time. Naked DNA means it's not bounded to histones. And DNA bounded to histones, or to say naked DNA is only found in prokaryotes. Or look at C, we know it's right. Let's look at D. Um, Cilias and flagellus are found only in prokaryotes. That's also wrong. You can also um, find these structures in eukaryotes. You can also find them in eukaryotes. Even though cilias and flagella are present in bacteria, that doesn't mean they're not present in eukaryotes, okay? But compartmentalization is found only in eukaryotes. That's correct. Your bacterias and more simplified forms of cells, they do not have compartmentalization. I can never say that word right. So yeah, the answer is C. I'll send you some link for practice questions, and you can have a go at them at home. If you have any questions, just send me an email or on the Crimson app, um, and I'll get back to you as soon as I can, okay? The homework is just to list out all the questions about the topic and send me one day before the class so I can prepare for them and better answer them and enjoy learning biology. See you next time.